Welcome to Biology, My Passion. I am Soumya Harikrishna. We have been dealing with the chapter Molecular Basis of Inheritance. We reached central dogma, and in that first process, replication, we studied in detail. Today, we are going to learn the next process in central dogma, that is transcription. So, what is central dogma? It is actually the way in which a gene is expressed. Expression of a gene is in the form of protein. But where is the information for making protein? On the DNA. So, prior to cell division, DNA multiplies or DNA makes an exact copy of it by a process called a replication. So, we learned replication in detail. Then, DNA is present within the nucleus in case of eukaryotes, whereas protein synthesis is happening outside in the cytoplasm. So, DNA cannot leave the nucleus and go out, and also DNA cannot directly synthesize protein. So, there is an intermediate molecule required, which is that RNA. So, first, information on the DNA will be copied into RNA. There are different types of RNA. I have done a separate video on RNA. You can see the link here. You can watch that video on RNA. Now, mRNA specifically will be coding for protein. Protein means what? Character. So, today we are going to learn this process. So, the formation of mRNA from DNA, that is transcription. So, we will learn that in detail. So, here also it has got certain similarity with our DNA replication, like principle of complementarity is governing the process. We know when a new DNA is forming, it makes use of the already present DNA to make the copy exactly like that. In RNA also the same thing happens. That is, based on the information on DNA, RNA is forming. So, if you look at the definition, the process of copying genetic information from one strand of DNA to RNA. So, isn't that a difference? Yes, because in case of replication, both the strands are copied, whereas here in case of transcription, only one of the strands will be copied. There are other differences also, we will discuss in detail. Look here, this is DNA, both are DNA. Here replication is shown, where the entire length of DNA is getting replicated. But when it comes to transcription, we do not want the entire length of the DNA to be transcribed. Only the part that is coding for a protein should be transcribed. That part is called a gene. So here is a gene. So this gene needs to be copied. That also, there are several thousands of genes on the DNA. All of them need not be transcribed simultaneously. So as and when it is required, they can be transcribed. Okay, so that is one difference. The whole DNA or the entire DNA is being replicated, whereas only a part of the DNA called a gene will be transcribed. That is one difference. Hope you understood. For example, if this is a gene coding for salivary amylase. Salivary amylase is an enzyme. Enzyme means what? Basically proteins. So only that gene needs to be transcribed. Understood? The second thing, here both the strands are being copied. So from one parent DNA, there is a new DNA forming. A new DNA also should have two strands. Whereas here from one DNA, what is forming? RNA. So from here, from DNA to DNA. Here from DNA to RNA. How many strands are there for RNA? Usually one strand, and specifically for mRNA, it is always linear. Okay, no folding occurs. So, and also only the mRNA, linear mRNA can give rise to a protein. Okay, so here only one strand is being copied. So that is the second difference. Only one strand of the DNA is transcribed, whereas both the strands of DNA are replicated. Now here comes another question. Why are both the strands not copied? Imagine these two are strands of DNA. One of the strands, the sequence, it's a hypothetical sequence A, T, you know, A stands for adenine, thymine, guanine, cytosine. Thymine, adenine, guanine, cytosine. So the other strand is complementary. So if adenine is there, opposite what will come? Thymine. Thymine means adenine. Guanine, cytosine. Cytosine, guanine, adenine, thymine, cytosine, guanine. Complementary. Now imagine if both the strands are making RNA. Are they going to make the same type of RNA? Let us see. So the first strand will be making RNA. It will have sequence similar to this because this is the complementary strand, right? So A will become U because RNA does not have thymine but it has uracil instead of thymine. So wherever T comes we have to make it U. Then rest all will be same. Similar to this. Now if this is making a strand, that will be similar to this strand. So here A, but T will become U, G, C, U, A, G, C. 3 nitrogenous bases constitute one codon or a genetic code for making amino acid. So here UAC, but here first one is AUG. Are they going to make the same amino acid? No. This will make one amino acid, this will make another amino acid. So from one gene, 2 different types of amino acid sequences will form, ultimately leading to 2 different types of proteins. But we want only one protein. For example, as I mentioned, if this gene is going to produce salivary amylase. So if one strand is giving the code for salivary amylase and the other strand also being copied, but it has information some for other enzyme, then what will happen to our metabolic reactions? Everything will become confused. Or imagine these 2 strands are like your parents. So the first parent, father is here, mother, 2 strands. Okay? Your birthday. They both are independently planning to give you a gift. They are not discussing each other. They want to give you a surprise. So father is going and buying a laptop. Mother is also going and buying a laptop. So as a birthday gift, what are you getting? 2 laptops. But do you need 2 laptops? They both serve the same purpose. You can use only one. So one is actually not needed for you. So in this case, if they both had discussed and bought one laptop for you, that would have been better. So here also, now you are confused which one to take. Or suppose they both are the same company, same model. It may happen sometimes, right? So it is confusing. Suppose you take one given by father, mother would become upset. If you take the one given by mother, father might become upset. So you don't want to hurt both of them. So this is what is happening when both the strands are being copied. They will make 2 proteins, but body does not know which one to use. It's a confusing thing, right? So always only one of the strands need to be copied. That is one reason. Another reason, from this strand formed, and from this also formed. But now look at their sequences. They are complementary. See, so these are all complementary strands. So what happens? They may go and base pair. As a result, a double-stranded RNA will form. From a double-stranded RNA, can protein be made? Absolutely not. So here, in spite of doing all this work, finally everything is becoming in vain because the RNA is pairing. So to avoid these confusions, both the strands are not being copied. So the first reason is, if both the strands are making RNA, they may code for 2 different proteins which will confuse the mechanism. Second thing, being complementary, the newly formed RNA may become a double-stranded RNA and it will not be able to go for translation or protein synthesis. Okay, now we will discuss other differences in this. So these are the differences between replication and transcription. DNA is formed, that is newly formed DNA, whereas here new RNA is formed from DNA. Similarity means both are forming from DNA. Then DNA polymerase enzyme is used. We learned DNA dependent DNA polymerase is the one making new strand. But here what is being polymerized? RNA. Here also depending on the information on DNA. So DNA dependent RNA polymerase is the enzyme involved in this process. Both the strands replicate. Here only one of the strands transcribed. We learned the reason. Entire DNA is replicated, whereas only part of the DNA or gene is transcribed. Primer is required. When we learn replication, we learned there requires a primer to start it, right? Whereas primer is not required for transcription. Deoxyribonucleotides are the substrate. Here ribonucleotides are the substrate. And one more difference if you want to say in that, thymidine is used here, here uridine is used. Then proofreading happens, here no proofreading happens. These are the differences between replication and transcription. DNA is a very long molecule. It has many genes, thousands of genes are there. But at a time, maybe a particular gene needs to be transcribed. How will it be demarcated from the rest of the DNA? So there should be a well-defined starting and ending point so that only that part is copied. Otherwise, the entire length will get transcribed. Okay, so if you see like that, if this part is the gene to be copied, it has a few sequences before that which is helping in the starting of the process and a few sequences after that to stop the process. So, a typical transcription region, a region which is going to be transcribed into RNA is called the transcription unit. A typical transcription unit has got three regions. A promoter region, the structural gene and a terminator. So promoter is for beginning, terminator is for ending the process, whereas only the structural gene is being copied. So if I add here a few sequence, this is promoter, here is the structural gene. DNA has two strands, but which one will get copied? So out of these two strands, they are anti-parallel. One is in the direction 3' to 5', the other one has the polarity 5' to 3'. So whenever we are considering a DNA, the strand with the polarity 3' to 5' acts as a template strand, or it is otherwise called an anti-sense strand or minus strand. See here we know when we talked about DNA dependent DNA polymerase in replication, we learned that a new strand forms only in 5' to 3' direction because new nucleotides can be added only to the 3' end, right? So here also it is the same. The new strand of RNA will form in the direction 5' to 3'. So which should act as the template? 3' to 5' because the new one forming will always be anti-parallel in polarity, right? Whereas the other strand, now what is the role of the other strand? It does not code for anything, right? But still it has got a name, coding strand. It's actually misleading. Coding strand does not code for anything, or it is otherwise called a sense strand or plus strand. Now naturally there comes a question. If it is not coding for anything, why should we call it as a coding strand? Because if you look at the first strand, I am writing only 4 bases for your understanding. Suppose here there is a sequence T, A, G, C. Okay? What will be the sequence in the complementary strand? T will become A, T, C, G. Now, if this is being copied into RNA, what will be the sequence on the RNA? T will become A will become actually thymine, but for RNA it is uracil, U, C, G. Now look at these two. They are same, only with the exception thymine and uracil. DNA has thymine, RNA has uracil. So the codes or the sequence in the coding strand is similar to that of the RNA forming. That is why it is called the coding strand. And also all the references we tell with respect to coding strand. So though it is not coding for anything, we are not completely neglecting it. It has to get some recognition, right? So for that what are we doing? We are trying to tell everything with respect to the coding strand. That is, if I am telling where is promoter located, promoter means what? The sequence which is helping in starting the process. So I will tell the promoter is located at the 5' end of the coding strand. I don't tell it is at the 3' end of the template strand. I will tell it is at the 5' end, or this side, left side is always promoter present. Whereas the downstream end or the right side, what is present? The terminator present. So the coding strand does not code for anything, but all references are made with respect to coding strand and its sequences are same like that of the RNA forming, except uracil in place of thymine. We have taken only the coding strand because we are now going to talk about the promoter. We know that promoter defines the coding, presence of the promoter defines the coding strand or the template strand. So if it is to the 5' end, whichever strand has it in the upstream or the left hand side, that is the 5' end, that is becoming the coding strand. So a scientist called Pripnov suggested that there are 3 regions in the promoter site. First, there is a recognition sequence. Here may be a recognition sequence where first the RNA polymerase will bind. Actually, it is coming from one end. Suppose this gene that we have drawn is here. Okay. So RNA polymerase is coming through the DNA, coming so fast, checking, and finally when it comes here, it will bind there. Okay. Then what happens? Next step is binding sequence. So after binding there, it will diffuse into that binding site. The next site is the binding site. So from the binding site, it will go to the next site, which is called the initiation site, from where the transcription can start. Okay, so once it reaches the initiation site only, the process of transcription will start. Suppose we consider this as the initiation site from where the transcription can start. Prior to that, there are 2 important sequences acting as promoters. The first one is 10 nucleotide upstream or left. So if this is taken as the starting point, left is considered minus. So if you count from here, 10 nucleotides left, that is called a minus 10. There, a consensus sequence is there. That is TA, TA, AT. Okay, so this is called a Pripnov box. So this is the name of the scientist who suggested it. Okay, so Pripnov box is present. Tata box also it is called, but we use Pripnov box in prokaryotes. So this Pripnov box is at minus 10 nucleotides away, that is upstream. Then again if you go further at minus 35, that means 35 nucleotides away from the initiation of the transcription, there is another consensus sequence TTGACA. If you look at the promoter site in eukaryotes, there are a few differences. So here also, if this is the starting site, 25 nucleotides away is Tata box present. Here, Tata box is called a Hogness box in honor of the scientist David Hogness who discovered it. So in prokaryotes, it was called Pripnov box. In eukaryotes, it is known as Hogness box. Okay, so they again after 75, that is upstream, 75 nucleotides upstream, there is another box with the sequence, but if you look only at this, it is called a cat box. So we call it as cat box, but the sequence is this. These 2 are the promoter sequences. But there is one more sequence far away from the initiation site, almost 1000 nucleotides upstream, that is in the 5' end or the left side, there is enhancer sequence. So that is not a promoter sequence. Promoter sequences are these 2 only, but enhancer sequence is a kind of sequence where special proteins can come and bind, because for the initiation of the process in eukaryotes, certain factors are required, protein factors are required. So this is providing a site for the proteins to bind so that the efficient activity of the enzyme RNA polymerase will be ensured. We discussed the promoter site in prokaryotes as well as eukaryotes. Now let us see the structural gene part. Structural gene is the part which is actually being copied into RNA, right? So we know that there are 3 types of RNA, tRNA, rRNA and mRNA. All these 3 types of RNA are equally important for protein synthesis. They have their own specific roles to do in protein synthesis or translation. So from where are they forming? All of them are forming from gene only, structural gene. So some part of a gene or some genes can give rise to tRNA, some genes can give rise to rRNA, some genes can give rise to mRNA. But we are now specifically talking about protein synthesis where RNA coding for protein is mRNA. So a gene, if you simply say, may refer to a part of the DNA or a DNA sequence which can code for any type of RNA. But we use a term called a cistron. It is a very specific term for a gene coding for only mRNA. Okay? So this is gene, this is gene, this is gene. But if I tell cistron, only this will be the cistron. Understood? So genes code for RNA, but the gene coding for mRNA is called a cistron. Only from that protein can form. Okay. So depending upon the number of cistrons, the genes may vary in eukaryotes and prokaryotes. In eukaryotes, they have very highly efficient system because they are very complex. So for them, one structural gene flanked between a promoter and a terminator is a single unit or a single cistron. Means what? It can give rise to a single mRNA. So single, the word is monocistronic. So eukaryotes have monocistronic structural gene. Whereas when we come to prokaryotes, for them within a promoter and a terminator, different cistrons are kept because they cannot have one promoter and terminator for each cistron. They are very simple. So many genes will be there or many cistrons are flanked between one promoter and one terminator site. So it is called a poly. Many means what? Poly. Polycistronic. Can remember P for P. Polycistronic is present in prokaryotes, monocistronic in eukaryotes. So this is an important differentiating question for you. Differentiate between polycistronic and monocistronic structural gene. And in structural gene, as we mentioned, two strands are there, which is the template strand, the one with the polarity 3' to 5'. So which is determining the polarity? We cannot say I can read 5' to 3' also. But what is determining the polarity? The presence of promoter. Promoter is present at the 5' end of the coding strand. So this is the 3' end of the template strand. Here is the coding strand. Understood? Another difference in the structural gene of eukaryotes and prokaryotes is that eukaryotes have split genes. Split means what? Separated, right? So here if you see, if this is the DNA, I have used two different colors. So these red ones are the ones coding for the mRNA. That means ultimately to protein. Whereas these are certain sequences which does not contain any valuable information. They are some repetitive sequences or intervening sequences. So these sequences which are coming in between the coding sequences, they are called the introns. Intervening sequences are called the introns. They don't code for mRNA. Whereas the red ones are called the exon. So here again exon, here intron. Here again exon. So only exons have the ability to make a protein. So what happens when mRNA is forming from a split gene in eukaryotes? It will have both the strands, coding as well as non-coding strands. So this is called a precursor mRNA or pre-mRNA. Now we have to process it. The eukaryotic mRNA formed initially needs to undergo some processing to become ready for translation. That we will discuss later. Whereas in prokaryotes there is no split gene. Okay? So what happens? The mRNA formed does not require any processing. It's ready for translation or the protein synthesis. If we see another difference is, in eukaryotes we know it has a well-defined nucleus and the transcription is taking place inside the nucleus and the precursor mRNA formed. Now it has to undergo processing before it goes out to the cytoplasm where the protein synthesis takes place. Whereas, so only after the transcription gets over completely, then some processing have to be done, then only it can go for translation. Whereas in prokaryotes, there are two advantages. First one, there is no processing required because forming itself is proper mRNA which can be translated. So translation can begin immediately. Second thing, there is no compartmentalization, there is no nucleus. So everything is happening, transcription as well as translation are happening in the same compartment. So what is the advantage? So suppose this is a RNA forming, by the time it is forming itself from one end, translation can begin. So transcription and translation are coupled in prokaryotes. This is an important question for you. Transcription and translation are coupled in prokaryotes. Give reason. First one, in prokaryotes, the newly formed mRNA does not require any processing before translation. Second, transcription and translation take place within the same compartment as there is no distinction between nucleus and cytoplasm. So these two can be coupled. Then with respect to enzyme also, there are differences. We will discuss that later. So now we finished promoter, structural gene. Let us move on to terminator site. So terminator site is located to the downstream. Downstream means what? Right hand side of the structural gene. Okay? Or we can say it is in which strand? Coding strand. Downstream of the coding strand, the terminator site is present. It is meant for termination of the process or the culmination or the stopping of the process. So here suppose this is the DNA from which RNA is forming. Okay, we will learn about that in detail. So once the RNA polymerase enzyme, which is actually capable of doing this process, once it reaches this site, so up to this is formed. Now hereafter it is the termination site. It is nothing but a sequence only. So when it comes here, it will start making a sequence again. But this sequence, certain parts are complementary. These sequences, then after that a few sequences will be there. Again another set of sequences which are complementary to this. So we know that RNA is actually single stranded, but whenever there is base pairing or complementarity, they can loop over. So imagine, suppose this end has a sequence GC, GC, then some other nucleotides, again GC, CGC. We know that these are complementary. So what happens? This RNA can fold and join back. So this GC, 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 this side what will come? GC, CGC. And this part which is not having a base pairing or complementarity will act as a loop. So this is the stem and this is the loop. So this is called a hairpin loop or stem loop structure. So once the RNA is completed, mRNA is completed, after that this hairpin loop will form. This is an indication for RNA polymerase to terminate the process. Okay, or there is one more way of termination, that is by binding with another factor called a Rho factor. Rho factor. When RNA polymerase binds with a Rho factor, it terminates the process of transcription. We discussed the transcription unit. Now what is the most important requirement for transcription? The enzyme RNA polymerase. So DNA dependent RNA polymerase. So this RNA polymerase enzyme can catalyze the process of transcription in the direction of 5' to 3'. That means the polarity of the new strand or RNA forming is 5' to 3'. Based on which template? The template strand with the 3' to 5' polarity. Then in prokaryotes, there is only one type of RNA polymerase that can catalyze all types of RNA formation, whether it is tRNA, rRNA or mRNA. But when it comes to eukaryotes, for each one there is a specific RNA polymerase present. For example, RNA polymerase 1 can catalyze only the transcription of rRNA or ribosomal RNA, whereas RNA polymerase 2 can catalyze the transcription of the precursor mRNA or the heterogeneous nuclear RNA. It is called a heterogeneous nuclear RNA because we learned it has got exons and introns. It has to undergo certain processing. Now third one, tRNA. The RNA polymerase 3 catalyzes the transcription of tRNA. 5, srRNA. SR stands for small ribosomal RNA and small nuclear RNA. So in prokaryotes, RNA polymerase consists of two parts. that is the hollow enzyme. The total enzyme should have two parts, core enzyme and sigma factor. Once these two combine together only, the RNA polymerase is functional, or rather it can catalyze transcription. Do you know why? Because if only core enzyme is present, it will transcribe both the strands. We don't want both the strands to be transcribed. So when it binds with the sigma factor, then it becomes a hollow enzyme. Hollow enzyme will transcribe only one of the strands. Now if you look at the structure of core enzyme, it has different subunits. They are alpha, two alpha subunits are there, beta, beta dash, delta, omega, omega 1 and omega 2 may be there. But it need not be present in all bacteria. Some like Escherichia coli have only 4 polypeptides, like 2 alpha and beta, beta dash. Whereas some other bacteria may have 7 types. So it varies. So what is the function? Beta is needed for the binding with sigma factor. So sigma factor can bind only at the beta chain. Whereas beta dash is required for the binding of RNA polymerase enzyme to the DNA template. And sigma factor also recognizes the start signal and directs binding of RNA polymerase to the promoter site so that the action of the RNA polymerase can start. So the first process of transcription is initiation or starting of the process. We know once RNA polymerase binds to the promoter site, the process starts. So RNA polymerase transiently or temporarily binds to sigma factor, then it becomes a hollow enzyme and it is functional. Now this will come and bind to the promoter site so that the process can start. Now if the DNA needs to be copied, we know that the DNA has to unwind, open, only then the sequence can be read and RNA can be formed. When we learned about replication, there were so many enzymes coming to help DNA polymerase enzyme. The helicase enzyme came and unbound it. Then topoisomerase came, made a nick and removed the tension in the supercoiling. So there were many enzymes involved in. But here there is single RNA polymerase enzyme has to do all this work. So once it is binding or coming to the promoter site, it will locally unwind the next region or the place where it is starting will be unwound. This is the starting point where our RNA polymerase is binding. Now what happens? When it is binding, it is now a hollow enzyme. This can unwind the next sequence, that is the next part of the DNA is locally unwound. So what happens is this part is open now. Okay, only this part is open, just like how origin of replication where opening happened, right? Y-shaped fork form, the same way here also it's opening up. So when it is opening, the sequences are now exposed. Suppose I am randomly writing a sequence G, C, C, T, A, T. Okay, so now what happens? This part is the complementary one. Suppose this is the template strand. Only template strand will be copied. So now the enzyme which is here will move through this and add the ribonucleotides according to the rule of complementarity. That is, if G is here, C will be added, G, G, A, then T won't come, U, U, A. Now up to this it reached. But here again, this is RNA formed. The template is DNA. So can they bind with hydrogen bond? No, they are two different molecules. So this RNA is only attached at a small part. Rest of the RNA form will be hanging down. Now meanwhile, when it reaches here, what happens? Now the enzyme is here. So up to this will close back. Okay, now it need not be opened. So this copying is done. So here the complementary strand is there. They will join back so that now the enzyme is here and this much RNA is here. Now the enzyme will move and copy. RNA polymerase reaches this side. The RNA is becoming longer. It will move further, unwinding, copying, joining back, again adding ribonucleotides till it reaches the termination site. Once it reaches the termination site, what happens? The Rho factor will come and bind with the RNA polymerase enzyme. Now, RNA polymerase will dissociate. At the same time, the RNA, which is newly formed RNA, which is bound to this enzyme, will also fall off. That is called a nascent RNA, newly formed RNA, that will also fall off. Now, by the time this is closed, sigma factor will already dissociate. Once the elongation starts itself, the sigma factor will dissociate. So, the final stage is termination by binding with Rho factor. That is the method explained in your textbook. There are 3 stages for transcription. Initiation, where the RNA polymerase enzyme binds with the sigma factor and initiates the process. Then elongation, the same enzyme will unwind the DNA and add ribonucleotides in a template dependent manner to produce the new strand of RNA, and this will elongate till it reaches the termination site. Then the last stage is termination. On reaching the termination site, the enzyme binds with the Rho factor. As a result, the RNA polymerase dissociates and also the nascent RNA falls off. Falls off means what? Where it is falling? It is falling down to the nucleoplasm. Right? It is inside the nucleus. So, this is the overall process of transcription. So, we discussed many differences between the transcription in prokaryotes as well as eukaryotes. What are the main differences? So, in transcription or in prokaryotes, RNA polymerase, a single enzyme is involved, whereas in transcription on eukaryotes, different types of RNA polymerase 1, 2, 3 are involved for the transcription of different types of RNA. Another difference we mentioned in the case of promoter site. Prignov box is present in the case of prokaryotes, whereas Hogness box is present in case of eukaryotes. In prokaryotes, the transcription as well as translation are happening within the cytoplasm. So, these processes can be coupled, whereas in eukaryotes, process of transcription is happening within the nucleus, whereas translation is happening outside the nucleus. And with regard to structural genes, we learn it is polycistronic in case of prokaryotes, whereas monocistronic in case of eukaryotes. Another difference is eukaryotes have split genes. What are split genes? They have exons of the coding sequences intervening with the non-coding sequences or introns. So, when mRNA is transcribed from that structural gene, the mRNA also has exons and introns. So, the mRNA needs to undergo certain processing to become the mature RNA. So, now what we got is the precursor of mRNA called the hnRNA or heterogeneous nuclear RNA. Now, it has to undergo processing to become the mature RNA. So, now let us discuss the post-transcriptional processing in mRNA of eukaryotes. So, this is the hnRNA we got with the exons and introns. Now, it will undergo a process called as splicing. During splicing, what happens? The introns, these green ones are the introns. Introns will loop out and will be removed. Now, exons are joined back. So, it is becoming a mature RNA with all the parts coding. So, you will get like this. Now, again, before it goes into cytoplasm, because this is now in the nucleus, before it goes into the cytoplasm for process of translation, 2 more additional processing happen. Which are they? One is capping, that is to the 5' end of it, methyl guanosine triphosphate, which is known as an unusual nucleotide. Okay, GTP with a methyl group attached to it will attach to the 5' end. That is called we can consider it as a head region, so it is a cap fitting there. Now, at the other end, a tail will form. What is the tail? It is simply adenylate residue. Adenine will be added, 200 to 300 will be there in a template independent manner, just a few A will be added. Okay, so if these processes are done, now the mRNA is ready to go to the cytoplasm and start the process of translation. Now, what are the functions of these two? They both are actually helping the mRNA from the action of hydrolyzing enzyme, or they will prevent the degradation of this mRNA. Also will help the attachment of smaller subunits of ribosome. So this is actually the transcription process in detail. Hope you understood the lesson well. If you like my teaching, please like, share and subscribe to my channel Biology My Passion.